All right, good afternoon, ladies and gentlemen. It is time to talk meiosis. I'm hoping that you have your meiosis puzzle with you. So whenever I'm talking about the actual stages of meiosis or events that are happening in meiosis, I'm going to recommend that you be adding notes to the diagram itself, to the puzzle itself. But when I'm talking about other things like the chromosomes themselves, then that could be like on a different page or in some other location. But if I'm talking about the stages of meiosis itself, why don't you write the notes right on your meiosis puzzle? All right, let's go ahead and talk about sex. Okay, no, it's not really about sex today, but we're going to talk about where sperm, egg, and in plants, pollen comes from. You guys, have you ever thought about that? Pollen is the same thing as plant sperm. Ew. Okay, so how do we get sperm, eggs, and pollen? Those are types of cells that are called gametes. They're sex cells, and they're unique because they have half the DNA. And I always say it that way because any cell that has half the DNA is called haploid. And any cell that has copies of both sets of DNA, so like you have 23 chromosomes from your mom and 23 chromosomes from your dad. So any cells that have all those pairs of chromosomes, 23 chromosomes, I want to make sure I said that right. Those are called diploid cells. But if you have a cell that only has half the DNA, that's a haploid cell. So that's what we're going to be talking about in this set of notes. All right, so first of all, what is sex? Well, just be aware that there are all different types of sex. So the way that mammals reproduce is not at all the same as the way many other organisms reproduce. And so you don't need to know any of this. Just I want you to see the integration or the rotation between the haploid and the diploid varieties of life. Our dominant variety is the diploid version, and our haploid cells are microscopic. But that's not true in all organisms. So it's kind of cool. Most organisms do not sexually reproduce. So bacteria, they just clone themselves. Hydra can just bud and produce a clone of themselves. Redwoods clone themselves by sprouting plants through their roots and growing a new tree. And then there's even something called parthenogenic reproduction, which is basically reproducing without a second partner. That one's a little bit more confusing. We don't need to talk about that one right now because that's not relevant to meiosis itself. So there are some background words that you need to know. So this would be an example of something I wouldn't write right next to your meiosis puzzle. This is background information, so maybe this goes on a different page or a different location. So fundamentally, all sexual reproduction involves the same cellular process. It's called fertilization. Fertilization is when the haploid cells of two organisms come together to make a diploid organism. Importantly, I want you to get the hang of the letter N. The letter N tells us how many chromosomes are in the haploid number. So we just say N. So for example, we are 23N. That's our haploid number. However, the diploid number is, I didn't say that right. We're not 23N. We're just N. Don't listen to me. Just N represents our haploid number. And then 2N, I'm saying it all wrong, 2N is the diploid number. So if, for example, we have 23 chromosomes in our haploid, 2N multiplies it by 2, and you have 46 in the diploid organism. All right, so take a look at the haploid and diploid levels for humans, fruit flies, and dogs. Fascinatingly enough, dogs have 78 chromosomes. Does that mean they're twice as smart as us? Almost 2/3 as smart, or whatever. Does it mean they're way smarter than us? No, not at all. Every species has their set number of chromosomes. Some of them are big, some of them are little. All that matters is that within the species, they have the correct number. So if I'm born with the wrong number of chromosomes, it gives me a genetic disorder always. There's always a syndrome associated, and oftentimes it's fatal. So having the wrong number of chromosomes for a human being is often fatal. Most of the time it's fatal. Okay, so sexually reproducing organisms need to make haploid cells that are called gametes. So that is a word you absolutely need to know. Gamete means sex cell, and it's always haploid. How do we make these sex cells when our cells are diploid? So how do we make those? And that's the process that we're going to be learning about called meiosis. In mitosis, the subheading on our notes on mitosis was non-reductive eukaryotic cell division. And that just meant that you start with a cell with 46 chromosomes, you end up with two cells that have 46 chromosomes. So we haven't reduced the number of chromosomes. In meiosis, we are reducing. We're going from 46 chromosomes down to actually 4 cells with 23 chromosomes. So that's why it's reductive. All right, oops, I'll back up just so you can see the image and kind of what we're going to be talking about here. So first of all, I'm going the wrong way, diploid chromosomes. So that's a funny way of saying it. So chromosomes in a diploid organism. Please, this is something that so sticks with people that they get it all mixed up. Don't mix up the term diploid with the term bivalent. So a bivalent chromosome is a chromosome that has two sides. It looks like a letter X. In fact, you should write this down in your notes right now. Draw a little letter X. So the left side is a sister chromatid, the right side is a sister chromatid, and this is called a bivalent chromosome. A diploid cell is a cell that has pairs of all of its chromosomes. So a diploid cell in a human being would be like a skin cell, has 46 chromosomes. Like my tongue cell has 46 chromosomes. Like my shoulder cell, 46 chromosomes. Those are diploid cells anytime that it has 46 chromosomes. You can have diploid cells that have monovalent chromosomes. You can have diploid cells that have bivalent chromosomes. You can have haploid cells that have bivalent chromosomes. You can have haploid cells that have monovalent chromosomes. The two terms have nothing to do with each other. Bivalent is just the shape of the chromosome. Haploid and diploid tell you whether there's pairs of them or not, whether there's two sets of X's or just one X. Okay, so it says here, remember that every diploid cell has two copies of each chromosome. You got half of the chromosomes from dad, and you got half of the chromosomes from mom. During S phase, each of these chromosomes is replicated. So then they become bivalent. So now you have a pair of bivalent chromosomes. A pair of bivalent chromosomes. So right now you could draw on your notes two X's. This is a bivalent chromosome. This is a bivalent chromosome. Now we have one from mom and one from dad. That's our pair. So the replicated copies, the two sides of the X, those are sister chromatids. So label each side of the X as a sister chromatid. But this X matched with this X from the other parent, that's called a homologous pair. The prefix homo means same. So it means that they're the same chromosome, but from different parents. And the word that they often use in AP or the College Board is homolog. It means the same as homologous pairs. It's just maybe a slightly faster way of writing it. So homologs and homologous pairs means the same thing. So that can be super, super confusing. So I want you to take a look at this picture here that's on the right. This is this black photo. That's a picture of somebody's DNA, and it's very confusing. So let's just talk you through it really quickly. At the end, we'll review this concept. But this photograph is called a karyotype, and it's a picture of somebody's chromosomes. What you're looking at, they redrew in cartoon fashion off to the left. And I think this is wickedly confusing because to me, when I look at that cartoon, it looks like an X. So I'm thinking, hey, KJ, that's a bivalent chromosome. Totally not a bivalent chromosome. In fact, that is two bivalent chromosomes. That cartoon picture, that's two bivalent chromosomes. One of them is leaning this way to the left, and one of them is leaning to the right. Look at them really closely. Look at the one that's on the right that's labeled sister chromatids. Do you see it's got two sides to it? That's because it's been replicated. So there are two copies and they're joined by a centromere. The other one is leaning the other direction this way, and it also has two sides to it. So it's just a coincidence that it happens to look like a letter X at the moment. If you look at the black photo, at the karyotype again, notice that they come in all different sizes and shapes. The one they chose, the pair of chromosomes they chose happen to be lined up so that they look like a letter X. But look at like chromosome number 10. One of the bivalent chromosomes is leaning off to the right. The other bivalent chromosome is standing very straight up. Look at chromosome pair number 16. One of the chromosomes is leaning off to the left, and the other one is standing up straight. So remember that they have the consistency of like a wet noodle or something, and so they don't stand up perfectly straight like X's or whatever. All right, so let's talk a little bit more about chromosomes to make sure you feel clear on this. So we're going to use red to represent the biological mother's chromosomes, and we're going to use blue to represent biological dad's chromosomes. So here we have a cell. All of the chromosomes have been replicated, which means that you started with a monovalent chromosome and then we copied it, and now you have 6 bivalent chromosomes. Notice at the very top where it says, how about now? And it says 2n equals 6. So that means the diploid number is 6. So if we look in this cell, we see that there are 6 chromosomes. 3 of them came from mom, 3 of them came from dad. That means the haploid number is 3. So when this cell eventually divides to form a sex cell, the sex cell will have 3 of these 6 chromosomes in it. All right, and then pause here if you need to, just to refresh your memory on some of the vocabulary that goes with this. All right, and I hate this diagram, so we're going to skip this. Like, just ignore this diagram because I find it extremely misleading. Okay, now is when we're going to talk about the actual stages of meiosis. And unfortunately, in this set of notes, they have it sideways instead of up and down, and your notes are up and down, but we can deal with that. So the first stage that you have on your meiosis puzzle is interphase. So remember that during interphase, whether that's before mitosis or meiosis, the DNA is getting replicated. So you might want to remind yourself, you could say S phase, DNA replicates. You could say monovalent becomes bivalent chromosome or something like that, just so that you remember that there's copies of the DNA during the S phase. So now the next stage, and the first stage they have listed here, is prophase 1. And let's talk about the first handful of stages. So prophase 1, metaphase 1, anaphase 1, and telophase 1. They're different, and you're going to have to write a lot of things about them. But the second half is almost identical to mitosis, so you won't have to write a whole lot of notes. So just be aware of that. I would write a note and then draw an arrow to the cell, and then write a note and draw an arrow to the correct cell. And then you should be able to fit in the ones prophase 2 through telophase 2 because you don't have a whole lot to write about them. Okay, so let's start by talking about prophase 1. All kinds of things going on here. So look at the sister chromatids. They have, and the homologous chromosomes. So during prophase, this is called synapsis, and they don't have this word written, I don't think, anywhere near here. So this is a word, you're going to want to write the word, you're going to want to write the definition and point to prophase 1. Synapsis is when the homologous partners find each other. Synapsis is when the homologous partners find each other. So one chromosome from mom, one chromosome from dad, they're the same size, they carry similar genetic information, but this one might say you're going to have blue eyes, and this one might say you're going to have brown eyes, but it's still the genes for eye color. They sit next to each other, and when they sit next to each other, we call that synapsis. Then the next thing that happens after they find each other is called crossing over. And now we're going to go into a little bit more detail here. So crossing over is when 2 bivalent chromosomes do this. They cross over and then they exchange genetic information. So that means that if this was mom's sister chromatid, so on one side of her bivalent chromosome, and this was the other side of dad's bivalent chromosome, they cross over, and then some of mom's DNA gets incorporated into dad's chromosome, and some of dad's DNA gets incorporated into mom's chromosome. Where they cross is called a chiasma. Plural is chiasmata. Oh, they do have the word synapsis here. I thought they skipped it. Well, it's right here. Okay, so chiasma is where they're actually crossing. So that crossing point. And then they're going to trade and they're going to separate. So it says, let's just read what it says. When chromosomes condense during prophase 1, so that means they twist up and now you can actually see the bivalent chromosomes, homologous pairs physically connect to each other during synapsis, forming structures called tetrads. At each connection or chiasma, DNA is exchanged between the homologous pairs. So it's got this very fancy name, crossing over. Crossing over is wickedly important because the DNA from your mom gets combined with the DNA from your dad into a chromosome that has never existed in the history of our planet before. It's a random process. And so every time crossing over happens, it's creating new variations of your mom and dad's genetic information. And that becomes really important because it increases diversity in offspring. And whether we're talking about your mom and dad or whether we're talking about a flea's mom and dad, we're increasing genetic diversity every time crossing over occurs. Okay, so here's sort of a little bit more information about it. So it says every chromatid, so that's, remember, that's a side of a bivalent chromosome, that is produced has a unique combination of DNA from both chromosomes in the pair. When they've crossed over, now the chromatids are unique. They're a combination of the mom and the dad's DNA. This results in every gamete produced having a unique sequence of DNA. Every single gamete. Every single one is unique. Oh my gosh. Sequence of DNA in each chromosome. This is called recombination. Now, we can do recombination naturally. This is recombination happening naturally. When scientists get involved in genetic recombination, that's what genetic engineering is when they're engineering and they're adding genes or taking genes away or moving genes around. That's recombination also. Okay, so let's just take a look at the diagram here really quickly. They have formed the chiasma. They are crossing over. And now look at this diagram. Notice that a part of mom's chromosome is now on dad's chromosome, and a part of dad's is now on mom's. And where they cross over, how much crosses over, how many times it crosses over is random. And so what ends up happening to the gametes is all this diversity in the gametes, and that's super important. Okay, moving on. So the next stage is metaphase 1, metaphase middle. What's super, super different about meiosis is that instead of lining up single file, instead of all 46 chromosomes lining up in one line in the middle of the equator of your cell, they're going to line up with their homologous partners. So they're going to line up in 23 pairs of chromosomes across the equator. So let's just look at that quickly. Homologous pairs of chromosomes line up at the metaphase plate, also called the equator, still attached to each other. When they separate during anaphase of part 1, the homologous pairs will separate. Sister chromatids will stay attached. What does that mean? That means that when we go to pull them apart in anaphase, they stay in their bivalent form. So the blue bivalent chromosomes are going to go to the left and the red bivalent chromosomes are going to go to the right. Be aware, don't be misled by this diagram, that it's by no means do all of dad's line up on one side and all of mom's line up on another side. This is another area where we can add genetic diversity. Mom and dad's chromosomes line up randomly on the right or the left side of the pairs. So one time you might have 23 of dad's on the left and 23 of mom's on the right. The next time you might have 10 of dad's on the left and 13 of his on the right, just to give you an example. Okay, and then let's see, I need to know if I need to click on this. Prophase 1, homologous pairs, blah, blah, blah. Metaphase 1, they line up as pairs. And then this is what I don't like about this one diagram. It says anaphase 1, but this is already telophase. The cell has not only gone through telophase, but through cytokinesis here. So it's a little bit misleading. But the homologous pairs separate from each other. But notice they remained as their bivalent chromosomes. So we're not quite to that yet. So I'm jumping ahead. Let's look at this couple of diagrams right here. It says this is a major difference. Metaphase is a major difference mitosis where chromosomes line up single file during metaphase. In meiosis, that doesn't happen until metaphase 2. So let's just sneak in big here. So this is mitosis that we learned originally. There would be all 46 of your chromosomes lined up in the middle. Then they get ripped in half. So the bivalents get ripped in half, and we have monovalent chromosomes in the one cell and monovalent in the other cell. In meiosis, they lined up with their partners, and now they get separated from their partners, but they remain as bivalent chromosomes. But now when the cell divides, notice what happened to the number of chromosomes. Here we had 6 bivalent chromosomes. Now we have 3 bivalent chromosomes. We cut the number in half. Well, why does the cell divide again if we cut the number in half already? Because these are bivalent. This is extra DNA. Let's divide the cell again, and we'll get 4 gametes out of it instead of just 2 gametes, because we had double the DNA that we needed in those. So that's what's going to happen. So that was metaphase. Now let's go to, we already kind of talked, oops, it didn't say anything more. Sorry, we already talked about anaphase, so just make sure that you've made notes that the homologous partners have separated from one another, that they stay in their bivalent form, and the note in telophase 1 that when the nuclei form, there's only 3 in this nucleus, or half the DNA in this nucleus and half the DNA in this nucleus. So this is where it becomes haploid. This cell, the initial cell was diploid, 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 diploid. Once they're separated, now the cells are haploid. They have half the DNA. Now we're going to go through the rest of mitosis, or meiosis, I'm sorry, but it's similar to mitosis. And the only reason we're dividing again is just to separate the bivalent chromosomes from each other. So there's no, it goes IPMAT, PMAT. There's not another interphase, so there's no more DNA replication. We've cut the number in half, and now we're going to go to prophase again. So prophase 1, the chromosomes show up. You can see them really clearly. Metaphase, sorry, I meant prophase 2. Metaphase 2, there's half of them. They line up single file like they did in mitosis. So there's no pairs. Why are there no pairs? Because their partner is in the other cell. So they can't line up in pairs. Their partner is in the other cell. So if this were a human cell, there'd be 23 lined up in the middle. And then in anaphase, we are going to rip apart the bivalent chromosomes. So monovalent chromosome goes to the left, monovalent chromosomes go to the right. Notice this diagram is really good that it shows that they've been recombined during crossing over. So notice the blue tip on this red chromosome. So we're going to add genetic diversity. And then finally in telophase 2 and cytokinesis, we're going to completely separate them. And what you need to understand is that these 4 cells, please write this down, the 4 cells at the end, they are gametes, they are haploid, they are sex cells. So you could say they're sperm, egg, or pollen. Those are probably good. And hypha, I think, is the right word, and fungi. I think that's the right word. So anyway, you write those down. Okay, meiosis at a glance. This is just kind of summarizing what happens in the whole process. Fundamentally, meiosis serves 2 major purposes: to produce haploid cells from a diploid organism and to create cells with unique combinations of genetic information. So remember, when meiosis becomes haploid, when the cell becomes haploid, that happens right at telophase 1. Now we have 2 haploid cells. And then the diversity comes in the order that the chromosomes line up, in the crossing over, and then also in which sperm and egg actually fertilize one another. The sperm fertilizes the egg, but whatever. When they get fertilized, it's random. Okay, and then let's just glance at this really quickly. You could spend some time looking over this if you wanted to, about the similarities and differences between mitosis and meiosis. They both replicate their DNA. Mitosis just has 1 division. Meiosis has 2 divisions. Synapsis, that's where the homologous partners find each other. That doesn't happen in mitosis, but it definitely happens in meiosis during prophase 1. Crossing over happens during that time, the chiasma, all that stuff. Okay, so let's move on, and then we're almost done. So we're just going to talk about why sex has evolved. So that's not entirely clear. However, there are some huge advantages to sexual reproduction. Let me back up. The advantage to asexual reproduction is that it is incredibly fast. So organisms that reproduce asexually, they don't have to find a compatible partner. And so they're just able to split in half or to bud or to do whatever they do and reproduce without having to find a partner. So that saves time and allows populations to reproduce more quickly. The problem is that they are, by and large, cloning themselves. And so the issue, and we're going to be really clear on this in our evolution unit, the issue is with a population that's been cloned, if the environment changes, if there's new climate, if there's new disease, if there are new predators, if there are new whatever, a cloned population is less likely to be able to survive those changes. Whereas a population that is extremely diverse, many of them might not survive those changes, but there might be individuals that can tolerate a new climate. There might be individuals that are camouflaged from the predators. There might be individuals that can survive the disease and have a better immune system. And so that increases that population's chance of surviving an event. And so they're less likely to go extinct. Okay, so in sexually reproducing organisms, one gender, the males, are not capable of producing an offspring. That's a handicap. So half of the organisms don't reproduce. And yet sex has evolved many, many times in many different ways. Okay, sex increases variation exponentially. Sexual reproduction leads to a tremendous amount of variation in a population. Asexually reproducing organisms only generate variation through mutations and horizontal gene transfer. So what they're talking about there is that if we have a group of bacteria, they're easy asexual reproducing organism, and all they're doing is cloning themselves, they should be all genetically identical. The only time that they become varied is if there's a screw up, which there often is, that creates a mutation. So a mutation might lead to a new branch of bacteria that are reproducing. The other way that they can get genetic information, this is not sexual at all, but sometimes they are able to do what's called gene transfer, which so some bacteria have the ability to actually create a tube where the DNA travels through from one cell to the next. And so this bacteria can give DNA to this bacteria. Some bacteria have the ability to absorb DNA that's in the environment. So let's say one bacteria dies and its DNA is spewed across the street. Another bacteria can suck up that other bacteria's DNA, and now they have whatever characteristics that bacteria had, they have that DNA. Those are examples of genetic transfer, horizontal genetic transfer. Okay, so take a look at the diagram here. This is sexual reproduction. All we're showing you is 4 chromosomes. Imagine the fact that you have 46 chromosomes. So look at what could happen. We could have the dad's chromosomes line up on the left and the mom's on the right, and this is what the sperm or egg would look like. This is their genetic diversity. Now let's see what happens if one of dad's is on the left and one of mom's is on the right. Oh, suddenly we have 4 combinations. So we have a total of 4 different possible eggs or sperm that could occur. Well, that's just with 2 chromosomes, ladies and gentlemen. You have 23 pairs of chromosomes, 2 pairs of chromosomes. You have 23 pairs of chromosomes. So let's do the math on that. I don't know if I'm going to need to be in the way. Humans, their haploid number 23, diploid number is 26. So independent assortment of homologous chromosomes. That's what it's called, and that's a phrase that you need to write down, and you should be maybe on a different page because we're not talking about specifically meiosis right now. I should have said that earlier. So right now, you need to know the phrase independent assortment. Independent assortment refers to which side the chromosomes in their pairs line up on. So one time when we go through meiosis, dad's chromosome might be on the left side and mom's on the right. The next time we go through meiosis in a different cell, dad's might line up on the right and mom's lines up on the left. It is random which side the pairs line up on, each one from the pair lines up on. So to mathematically calculate that, it says here each homologous pair has a 50-50 chance of lining up so that mom's pair or dad's pair winds up on either side of the cells that are produced. Mathematically, this means that there are 2 possibilities for each of the 23 tetrads. So what do you do for the math on that? 2 to the 23rd. So back up, look at this. This one with just 2 pairs of chromosomes was 2 to the 2 raised to the second power, so 4 possible combinations. But we have 23, so ours is 2 to the 23rd. So 2 to the 23rd is over 8 million possible combinations of our gametes. So any sperm or any egg, any human that's producing them, there are more than 8 million combinations of the DNA. That's just one way that we can add genetic diversity to our sperm or egg, is called independent assortment. Another way that we add genetic diversity is through crossing over. And random fertilization. Let me do it in reverse order here. So random fertilization. So we never know which sperm and which egg are going to combine. And if dad's sperm had 8 million, there's more than 8 million possibilities, and mom's sperm has more than mom's eggs, I'm talking too fast. Mom's eggs have more than 8 million possibilities. You multiply them together, and for any given offspring, in at least a human being, there's more than 70 trillion possibilities for the baby in one family. What? So in essence, we're going to say that genetic diversity within a human population is virtually infinite. That's crazy. There's one more thing that increases our genetic diversity that we already talked about, and that's crossing over. So in this previous diagram right here, notice that there's no crossing over. So there's no, this blue chromosome is the same here, it's the same here, and it's the same here, and it's the same here. But once we add crossing over into the mix, none of the chromosomes are ever the same. And so we've just added more diversity. So in essence, we say that diversity is functionally infinite. We have so many different ways of producing offspring. Okay, we're almost done. The last thing that we're going to talk about just quickly is the karyotypes. We will learn a little bit more about karyotypes, and you will learn to analyze karyotypes later on. So karyotype is a photo or a drawing or a compilation of somebody's chromosomes. So they look at the chromosomes under the microscope. So this is what they might look like before they're sorted, and then they organize them by homologous partners. So they organize them really by height. So the tallest 2 chromosomes are called chromosome 1, and so they're always the tallest 2. So chromosome 1, there are 2 of them. One you got from mom and one you got from dad. So look at it right here. This one maybe you got from mom, and this one maybe you got from dad, and this is the homologous pair of chromosome 1. So both of those are chromosome 1. Chromosome 2, notice this one's bent over. Who cares? It's like a limp noodle. It doesn't matter that it's bent over. So the rest of these are the homologous pairs. What we are going to look at in the future is when we have the wrong number of chromosomes, or when one of the chromosomes is missing some information or has extra chromosomes, has extra information. Couple things to just learn, couple vocabulary words. Autosome, so just like the word chromosome, and autosome are the chromosomes that males and females have equal. So we have 22 chromosomes that are the same, whether you are biologically male or biologically female. It's not until we get to the last pair that determines at least biological sex. And we're going to learn there are infinity combinations of things that can happen to the sex chromosome. So the sex chromosomes are always listed last. There's the X chromosome and the Y chromosome. Notice how tall the X chromosome is compared to the Y chromosome. Females get 2 copies of the X chromosome. Males get 1 of the X and then 1 of these small Y chromosomes. It is possible to have the wrong number of sex chromosomes. There's all kinds of crazy things that can happen with sex chromosomes. Super interesting stuff. All right, and so these are just normal karyotypes for a male and a female. So here's the male, it has an X and a Y. Here's a female, it has 2 X's and no Y's. So you should be able to look at a karyotype and be able to identify whether they are biologically. Now that has nothing to do with gender, keep in mind. Gender is more a social construct, how we feel about ourselves. But in terms of the biology about whether, and even that is complicated. And we can talk for hours on all of that. But biological sex, XY male, XX female, but there are even variations on that. All right, cuties, that is it. Have a good rest of your day. Oops, what am I doing? I'm going to have trouble stopping this now.